can you start by telling us your name, title, and institution?
Yes, my name is Greg Mohl. I'm a postdoctoral fellow at the University of California, San Francisco. I also am an Atlantic Fellow with the Global Brain Health Institute.
Can you provide a brief overview of your SCGE project and explain your role within the project?
Of course. So, our SCGE project, I work with Dr. Claire Cleland also at UCSF. And our goal is to develop gene editing therapies for genetic neurodegenerative diseases. And the gene that we're focused on right now is called C9orf72. And there are these repeat expansions in this gene that are the primary genetic cause of genetic ALS and FTD. And so by developing this new therapy, we should be able to hopefully cure or treat people who have genetic ALS and FTD caused by C9orf72. Yeah, my role, I'm focused on delivery. And so Dr. Cleland has done a lot of work in understanding the biology of C9orf72 and like developing gene editing strategies that will be the most effective. But the primary challenge for gene editing right now, especially in the brain, is delivery, getting the therapy to the right parts of the brain safely and at enough of a scale to have a meaningful therapeutic benefit for patients.
Okay, great. And you did just touch on this, but can you explain to people what are some of these challenges of delivering CRISPR therapies to the brain?
Yeah, of course. So this is not a new problem. This is something people have been grappling with for a long time. In the early days, people tried just directly injecting gene therapies into the brain. And the problem is that in the brain tissue, most um, gene therapy vectors or carriers do not diffuse very well. And so what you end up getting is wherever you injected it, you get really high delivery to that tiny spot, but you aren't able to deliver it far and wide, which for most of these diseases is what you need. So sort of the holy grail or the ideal situation would be to cross the blood-brain barrier because almost every cell in the brain is 10 to 20 microns away from a blood vessel. So if you could cross the blood-brain barrier, then you would basically immediately be able to cover most of the brain, essentially all of the brain. The blood-brain barrier is uh, an aptly named. It's a barrier and it exists to prevent stuff from getting into the brain, and it's pretty good at its job. Um, so those are sort of the challenges. And we actually have partnered with a team at the Broad Institute who's also a part of the SCGE from the Deverman Lab. And they've developed this new AAV virus that is actually very efficiently able to cross the blood-brain barrier. And so we're kind of using that as our first generation version to try and get to the clinic and get to patients as quickly as possible.
So, how is your team working to address these challenges and improve delivery of gene editing therapies to the brain?
Yeah, yeah, that's a great question. So, like I said, for the first generation therapy, we're partnering with the Deverman Lab at the Broad. And again, that's just because we're trying to get to patients as quickly as possible, which is sort of the impetus of this consortium. But we're also thinking long term, and that's really what my project is focused on. And so we're building platforms to screen large libraries of nanoparticles, including AAV, but also lots of non-viral delivery technologies in a pooled format in lots of different model systems. So we're using mice, we're using non-human primates, but we're also moving towards using human platforms to actually get human delivery data with these large libraries and try and unlock some of these hard to deliver to tissues and cell types. And yeah, I'm very excited about some of the new technologies that are coming out, and I'm hopeful that using these types of approaches, we can accelerate um, the timeline for getting these new therapies to patients for many, many different diseases.
So, how do you balance designing these effective, optimized gene editing therapies with the time pressure to get these therapies into clinical trials and to patients?
Yeah, this is, I think, one of the great challenges. Is, we can tinker away forever and improve the delivery, improve the editing efficiency, and at some point, you can't let the perfect be the enemy of the good, I think is the common saying. And so, obviously, there's a lot of challenges with using AAVs, um, especially in the brain. And so I think in the short term, it makes sense for us to use this technology because it's the most advanced technology and it'll enable us to rapidly deploy and hopefully start saving people's lives. Um, so that's kind of how we've balanced that is by kind of using what's available and what is likely to work in the short term, while also building out capabilities to enable, long-term flexibility and sort of long-term planning. I think that's how we've balanced that is by just trying to move quickly.
The next 10 years, what is something that you predict or hope will happen in the gene therapy field?
Yeah, that's a great question. I think, like many of us in the field, we were really excited by the story from of baby KJ from the team at CHOP. For those of people who don't know, it was actually an SCGE team that developed the first ever in vivo um, custom gene editing therapy for a baby that had a urea cycle disorder. And what I'm really excited about is the potential for these types of custom therapies to become more accessible and available to patients and also cost effective. What I really hope happens is that it becomes something like a surgery or a transplant, where it's a routine procedure you could get at your local hospital where you get sequenced, you identify a mutation, they can quickly make the therapy for you, and it doesn't cost $1 million. So in all of the work that I'm doing, I'm trying to think about, because there's all these cool ideas that I have, but I'm trying to think about like if we were to actually try and get this to patients, is this going to be cost effective? Is this something that's scalable and that would fit in this vision of like trying to cure as many people as we can? So, I both predict and hope that this happens in the next 10 years in the field.